Hello, everyone. My name is Max Norman, and for my faculty project, I work with IQVIA. So for those of you who don't know IQVIA, they're the global leader in pharmaceutical consulting. And I work with them to predict clinical trial failure due to toxicity. So before I start this presentation, I would ask you, picture yourself as the CEO of your very own pharmaceutical company. And your lead scientist has approached you with a groundbreaking treatment which will solve, say, cancer, diabetes, and dementia all at the same time. Now, before you can go saving millions of lives and giving hope to millions of patients, your treatment's going to have to undergo a clinical trial. So what is a clinical trial? Well, clinical trials are a fundamental part of drug development, and they're typically split into three phases. Phase one focuses on pharmacokinetics, and so this is how the drug interacts with the body, and how it moves inside, and the safety of the drug. Provided it passes this phase, we'll move on to phase two, which evaluates the efficacy of the drug. And finally, onto phase three, which will evaluate whether the clinical trial succeeded in meeting its endpoints. The details aren't too important here. I'll only ask you, there are three key takeaways. Number one, they are very long. There's a lot of financial and time investment that goes into these clinical trials. Number two, there are a lot of participants involved, which means that before we even know the approval status of this drug or this treatment, we're giving it to a lot of participants. We're giving them hope that their treatment will be effective. And finally, the most important, very, very few drugs actually make it to phase three. Very few drugs complete their clinical trial. Now, why is this? There's a number of reasons, but for the purpose of this project, I focus solely on toxicity. Toxicity being any adverse event caused through drug use, either at the therapeutic or non-therapeutic doses. The reason I focused on toxicity is because this is an observable phenomenon that can be studied with a scientific method. The enrollment and funding reasons are more business-oriented, and as a result, don't present the same opportunity to be able to leverage the power of AI. So at this point, I'd like to remind you of your very important roles as CEOs of your very own pharmaceutical company. In this point where your lead scientist has just presented this groundbreaking treatment, wouldn't it be fantastic if at the snap of a fingers, you could know whether your drug or this proposed treatment, before you've invested all of this time and money, before you've given these patients the hope, whether it's going to succeed in its clinical trial. And this is exactly what I did. So with IQVIA, I basically looked at taking a proposed treatment for clinical trial, identifying the key target and structural features before beginning the clinical trial, using those features to develop a predictive model to then make my predictions. And these key target and structural features, after some experimentation, were found to be the molecular structure, which I'll touch upon in a minute, targets, so specifically the hERG protein, which is an important and fundamental protein in normal functioning of the heart. So you can imagine that a drug which acts as an inhibitor to this protein, which effectively means stopping the normal functioning of this protein, can cause serious cardiac concerns. Secondly, the P-glycoprotein is a fundamental moderator in drug-drug interactions. So again, inhibition of this protein can have disastrous consequences. Finally, we took drug likeness rules, which are essentially developed by practitioners within the industry to try and group similar molecules based on their structural properties. So now touching on the molecular structure side of things and going a bit deeper, we're all very familiar with the structure presented on the leftmost of the slide. This is what we're presented with when we study chemistry, for example, or biology in our high school days. Unfortunately, although this is very intuitive for us and it gives us a lot of information, this is useless for a machine learning model. So to make this useful, the first step is to turn it into a simplified molecular input entry system, commonly abbreviated as a SMILES. And so what this does is it basically reduces the two-dimensional structure that we see on the left into a sequence of characters. And what's great about this is that we've reduced 2D to inline. However, although this is an important step, we're not there yet. What needs to be done is we need to find a way to turn this SMILES into something numeric. And to do this, we use its extended connectivity fingerprint, also known as ECFP. And so what this does is it basically creates a numerical representation of the molecule we have on the left side, but it's uniform, which means that we can do this for every single molecule known to us in the small molecule side of things and pass it into a machine learning model. So on that end, we take the aforementioned features combine it with this molecular structure feature, and build a deep learning model to predict the probability of clinical trial failure due to toxicity. And now, how well did we do? It turns out pretty damn well. And the key, important, fundamental thing from this project is that molecular structure is a key predictor of toxicity. So much so that just including this molecular structure into a model increases the performance, increases the accuracy by roughly 20% above the current industry benchmark. What's more is that incorporating this molecular structure, we can see in our 100-fold cross-validation that the same model trained without this molecular structure information is a lot less reliable. So the sharp dark blue peak that you see on your screen essentially means that we can be very, very confident in our predictions, as opposed to the more spread out and blobby version where no molecular structure was considered. And so what is the impact of all of this work? Well, primarily, we're improving patient safety. At the heart of it, toxicity events are recorded in clinical trials when a patient has such an adverse effect that they need to be hospitalized. So if we can screen drugs which are likely to create these effects in a clinical trial and stop them from ever going forward, then we're improving the safety of the patients participating in those clinical trials. And what does this lead to? This leads to a better overall treatment. And finally, as important CEOs of your very own pharmaceutical company, allocations of resources is a very important topic for you. You do not want to be investing millions of dollars and years into a clinical trial to find out at the very end that you failed due to something that I could have predicted from the beginning. And so this brings me to the end of my presentation, and I'd like to thank you all for listening.